Again, the goal of our project is to see if we can figure out which factors lead to someone becoming a regular smoker versus a non-smoker. And so as part of this, we took a look at the dopamine pathway and some of the molecules involved in that pathway. The reason we looked at this pathway is because there's a chance that the genes that encode for some of these molecules might influence whether someone becomes a regular smoker or not. So for example, someone might say, you see this molecule, dopamine decarboxylase, I wonder if the gene that encodes for it, I wonder if differences within that gene is one of the things that influences whether or not someone becomes a smoker. So in order to figure that out, you'd first have to know what particular version of the Ddc gene a smoker has versus a non-smoker, so you can make that determination whether it has an influence on that. So the term used to describe the process of determining what version of a gene someone has is genotyping. So we'll spell it out in words here. Again, the process of determining which alleles an individual possesses for a particular gene. And it turns out that as part of this project, some people at the University of Washington have actually already genotyped several people for their Ddc gene. And so basically it's going to be up to us to look at that data and figure out if there is any correlation between differences in this gene and the susceptibility of becoming a regular smoker. Here's some information on the Ddc gene, and actually here's some information on one particular portion of the dopamine decarboxylase gene. It turns out that there's a polymorphism within intron number 9 of this gene. A polymorphism that involves some people having a G at a particular portion within intron 9 and other people having an A within a particular region of intron number 9. Here's what we mean by that. So if you look at the Ddc gene, this gene has several introns in it. And what we're saying is that if you consider just intron number 9, some people have a version of intron number 9 that if you take a look at its sequence, there happens to be a G in a particular portion within it. Another version of intron number 9 of the Ddc gene has this. If you take a look at its sequence, it has, instead of a G, it has an A in that particular portion. So by this description here is saying that in intron number 9 there's a single nucleotide polymorphism changing G to A. We're saying that there's two different versions out there for intron number 9 of the Ddc gene. One version has a G in a particular region, and another version has an A in that corresponding region. And so if you're going to describe the possible genotypes that someone can have for intron number 9 of the Ddc gene, here are the possible genotypes that a person can have. Right? So it's possible that someone inherits the G version of intron number 9 from both parents. So another way of saying that is that the person has the GG genotype. It's possible that someone would inherit the A version of intron number 9 from both parents. And so if that's the case, that person is said to have the AA genotype for that particular region. It's also possible for someone to be heterozygous for that region. So if that's the case, that means that from one parent he inherited the G version of intron number 9, and from the other parent he inherited the A version of intron number 9. This is the case for anyone, so these are the three possible genotypes that someone could have for the 9th intron of the Ddc gene, for both smokers and for a non-smoker. And so what we're going to do, actually what genotyping did, was that it allowed us to figure out exactly which genotype each participant in the study had. And the way that this was done was by doing something called a reverse dot blot. And so in order to do a reverse dot blot, what's required is first you need to make many copies of a person's intron number 9 in order to have sufficient DNA to study. And the way that many copies of this DNA was obtained was by performing polymerase chain reaction, which we actually did in the lab earlier this quarter, and which we described in class. So if you're not familiar, or if you don't remember how PCR works, it's probably a good idea to go back and review your notes, or take a look at that video, that tutorial online that reviews how PCR works. But what we'll do here is take a look at how the reverse dot blot worked for this particular situation for our project. So again, what we were interested in doing in our project was to determine what version of intron number 9 the participants had. And the intron number 9 that we're talking about is one that's found in the Ddc gene. So remember that we have, all people have 2 versions of the Ddc gene, represented by these green stripes. And again, that's the particular gene that we were interested in this study. So let's focus in on those 2 chromosomes that have those genes and blow them up, enlarge them. Specifically, we were interested in just one of the introns within these Ddc genes, intron number 9. And so again, in order to figure out what version of intron number 9 this person has, we first had to make many copies of intron number 9 using PCR. And so if you remember, the way that PCR works is that you throw the template DNA and some other stuff into a test tube, and first, in order to copy these DNAs, you have to separate the 2 strands from one another, kind of like this. Once the strands have been separated, then the primers are allowed to bind, and in this case we use primers that bind to just either end of intron number 9, so that we can just get many copies of intron number 9. So we use primers that did this. And it turns out that the primers that were used in this particular project were kind of special primers, because not only were they primers that allowed us to copy just intron number 9 of the Ddc gene, but the primers that were used had a molecule called biotin that was attached to it. And so in a bit we'll see the reason why primers were used that had biotin attached to it. And so if you're kind of continuing on with looking at what happened during the PCR, after the primers bound, then the polymerases were able to attach nucleotides to the 3' end of that primer, and that happened until it ran out of template. So it did that to all of them. And so again, keeping the big picture in mind, what we're trying to do is just make many copies of just intron number 9 of a person's Ddc gene. If you look here though, after just one round of PCR, you have not only intron number 9 that's being copied, but a whole bunch of extra stuff that you don't want copied that's outside of intron number 9. But remember that if you let PCR continue for a few cycles, you'll eventually get just the region that you're after. So to kind of review that and to visualize that, about if we get rid of those duplicates here to simplify and see what happens next. When another round of PCR initiates, it involves separating the strands from one another again, allowing the primers to bind, and again allowing the polymerases to add nucleotides to the 3' end of each one of the primers. If you do another cycle, same thing will happen. The strands will separate, primers will bind, and then the polymerases will add nucleotides to the 3' end of each one of these primers. So what we're saying is that if you let enough PCR cycles go, eventually you'll get copies of DNA that just have intron number 9 in it. So we'll get rid of then the other extraneous stuff and focus in on the stuff that we're after during the PCR, which is just a bunch of these guys. And again, because the primers had biotin to it, that means that all of the intron number 9s that are produced are going to have 2 biotin tags to them.